Okay, so in this video we're going to be going over where to find the COVID-19 reference sequence. It's located on the NCBI website. Then we're going to explore a few tools on how to kind of compare the sequence with other sequences. And then we're going to go over looking at it in Python and seeing what that looks like. So let's jump into it. We're going to jump into it by going to the NCBI website, just a home page. And then we're going to navigate on the left hand side. There's going to be a genomes and maps link right here. And when that loads up, there's going to be a tab on the top that says database. There's going to be a bunch of cool databases in here, but the one that we're looking for is called SARS/CoV-2, I think. Oh no, just the SARS-CoV. There it is. So right here, what you're seeing is that it's already filtered for what you're looking for. If you're looking for COVID-2 here, you see if you undo the filter, you'll get like 10,000 reference sequences. So you want to make sure that your filter's set up for the proper sequence, and there you go. Let's go ahead and download it now. So the top assembly, the one that's .2, this is the one that we're targeting for download. And we're going to go ahead and click on that top reference assembly, and then it brings you to this page where you'll scroll down and you'll see that NC underscore actual number, and then you got yourself the Wuhan here. And FastA on the top, we're going to go ahead and click on FastA, send to, and file. Make sure that everything looks like that. Choose, and there you go, it's downloaded. You have a FastA file. But while we're here, let's go ahead and take a look at what all this information means on the NCBI website.
Okay, so real quick, while I got a moment here, I'm going to go over this file. So this is your nucleotide base pair file that you're going to see when you run your print on the FastA file in Python. So the screen right before this file, though, has a lot of great information on it and kind of talks about how the file's aligned. So shout out to these great authors right here. Keep up the great work. And right here where features and sources start. So you got your 5' UTR gene. So between the first position of the file and then the next 265 positions, that's going to be your 5' UTR gene. Then you have your coding sequence, and now basically like an open read frame. They say that all coding sequences are open read frames, but not all open read frames are coding sequences. And then you have your peptide. Let's see, we're looking for gene. Gene, those are the fun ones. Okay, so we have where we're starting our first gene. So not only is this the first gene in the sequence, but this is the spike glycoprotein gene that, you know, was all the bee's knees in the coronavirus. This is what people were really targeting. And as far as our sequence goes, this is the location of it. And then you have some more coding sequences and into more genes. You got some open read frames. And if you want to know more about the genes and what and their impact, what I highly recommend is reading a Nature information. So if you go and type in COVID-19 RNA sequence, I looked it up right before. So that's COVID-19 RNA sequence. And you go to this genome-wide analysis. There is a nice chart right here, and it kind of shows you what you're looking at. So if you take this as your gene that or not your gene, these are many genes. If you take this as your sequence, that is actually right in this file right here, in this FastA file, this is actually your sequence in a picture format. And you have your genes locations here, A, B, C, D. And you kind of have what their alleles are and some studies with differences of alleles in this particular image. But they kind of overlap with these genes right here. So you got your spike glycoprotein gene, and you can kind of match them up, the genes, with this graphic. and that's kind of what they mean. So, and then you got your positions. So let's go ahead and zoom in a little bit here. First, let me. So what I'm going to do here is I like working with the important stuff, and we got our glycoprotein. What we could do here is we could just click on any of these areas. If you click on them, they'll actually highlight the area. So we have our spike glycoprotein gene. This is it right there. So now let's just ensure or use another tool just to compare. I just want to show this other tool that I've been using here. I'm going to put the link in the description. Shout out to the University of Virginia. This is a real awesome tool. So first, we're going DNA to DNA or FASTA. There's a lot of different formats, but it's nice and easy for us. We know we got a FASTA file. What we're going to do here is let's go to the actual FASTA file. We could use this area right here. You could use any area of it. But just to conceptually see, we're going to go ahead and just take the first row of this and sort it to viral and then search the database. Now there's a lot of options here, and you can compare different strands of the virus. So remember, there are other strands, there are other sequences that you can compare. So this is a great tool that you can use to quickly compare, try to filter out things that you want to look at maybe in your Python searches. But let's go ahead and hit search database here. It takes a few seconds, and then the search is done, and it really only takes about 5 seconds, and then you get yourself a bunch of information about the sequence you just searched. I'm pretty much certain now that we do have the coronavirus. There's our spike protein right there. It sensed that that's an associated sequence with what we put in. So now let's jump into trying to read it in Python so we can use comparing tools that you use in Python. Okay, so I have some other videos on how to really view these FASTA files, but I really wanted this one just to be how to get it quickly up on the screen. So if you open up, I use pycharm, and this part already kind of requires you to have a knowledge of Python. So I'm going to make a new, I didn't pick an interpreter. Great. I'm going to have to pause this. Okay, so I got it going again, and really you want to make sure that you know where your location is for your file, and then you want to go get that sequence we downloaded earlier. So I went to my downloads area in my Google browser. I went to show in folder. I'm going to grab this sequence right here, and I'm going to bring it over to my Python pycharm screen. And this is the directory that I'm using for that I want my file to go to, and it's as simple as just dragging it in here. And now we have it showing in Python. We have our sequence FASTA, and we can call it in our main Python page. So if you want more information on how to search it in or search this file in Python, I have other videos on how to search a FASTA file, and in the future here, hopefully early 2021, I'm going to be putting out a video on how to compare this virus to other viruses from our history using information off the NCBI website. They have many other virus sequences that you can compare this virus sequence to, and I'm going to be using some other Python tools and code to help me do that in those videos. But that's it for this weekend. This is just a quick how to get the file so that I can set up the other videos. And if you want more information on how to search this FASTA file, check my earlier videos out, and have a good weekend.